put into your semester project report has to be documented. You cannot make factual statements that aren't facts. They can't be alternative facts. They have to be facts backed up by reference. So in this case, salmonella can cause gastrointestinal infections. How do we know? Because Gold published a document in 2006 that said they did. They're sometimes found on pet turtles. Who said that? Ying in 2008. Pet fish have been found to carry Escherichia coli, another type of pathogenic bacteria. Who said that? Abera in 2009. So whoever wanted to test this water, they would do some research and find out what kinds of things they might expect to find, and then they'd come up with a hypothesis. The hypothesis is what they think they might find in the water. So they can say, cultures obtained from turtle and fish tank water are likely to include salmonella and or Escherichia that are potentially pathogenic. That is the hypothesis, okay? This is not a master's thesis you're gonna be working on. It's pretty short, but your introduction should have multiple references. The other thing is you wanna do your research, because if you were gonna work with Escherichia coli, you need to know, what does it look like? You take a sample of water, what are you looking for? What do you expect to see? If you see colonies on a plate that are bright purple or bright pink, that's not Escherichia coli, because they don't do that. If you put them on TSA, the colonies are just kind of white or gray, off-white. They're not exciting looking. So you want to learn something about what you expect to find. Now, each person, I would expect to have one organism type that they're going to isolate. So if you're working in a group, let's say a group wants to do fish tank water, you want to identify several different organisms that are in that fish tank. Each person selects one organism type, and that's going to be their unknown that they're going to work with. So one colony per person. You don't have to work in groups. You can work independently. Each person has one organism type. I don't want to have groups and have 5 people with only one organism, because if it dies, you're up a creek. So each person has to have their own organism type, their culture. So you take your sample, again, we're using very artificial techniques, but we're growing these organisms in pure culture, so you're gonna put them on some kind of medium, either Nutrien Agar or TSA. You have to establish a pure culture before you can do the identification. That's just because of the way we do it. They must be pure. Why? Because we're gonna take the DNA out of them and we're gonna sequence it. We're gonna amplify it with a PCR, we're gonna take it down to Davis and sequence it. If you have 2 organism types, you've got sequence on top of sequence and you can't read it. It's worthless. They have to be pure cultures. Once you have your pure culture, you establish the cultural characteristics, again, colonies and cells, except that you don't do all the stain techniques we've done for the morphological unknown. In fact, you only do 2 stains. Indirect, and it can either be nigrosin or Congo red. Our indirect that we did with the morphological unknown, that was nigrosin. Tonight we're going to do a capsule stain. You can use Congo red if you want to, so you can compare how they work. But you would just do those 2 stains, indirect and gram stain. The gram stain tells us what kind of wall the culture has, and we need to know. You can also do a KOH, because a lot of times a KOH is more accurate than a gram stain. We have some bacteria that stain pink, but they really have gram-positive type walls. And we need to know that because we have to get the DNA out, and the processing is different. Once you determine what the wall type is by doing a gram stain and a KOH test, then we take the DNA out and we do a PCR. And as I said, that then goes to Davis for sequencing. So we need to get started. The price for sequencing, the price is lower if we have 48 or more samples. There's 30 people in here, but oftentimes I can combine ours with ones from Warren, so we can get 48 or more fairly early in the semester. We can get that DNA to Davis and get our sequence information back early in the semester, which we want to do. So you need to think about getting started on your semester project ASAP. You know how to streak plates. You know how to isolate and get pure cultures, I think. I hope. Now, back to our morphological unknown. Your morphological unknown, you have 2 additional steps you have to do. You're going to prepare a capsule stain. This is on page 7980, I think. And and you're gonna prepare a wet mount and determine if or not your culture is mobile. Neither one of these should be extremely time consuming, so I think we should be able to get through this and still have time to do our introduction to prokaryotes. This is introduction to the capsule, the glycocalyx. Again, it's a material outside the cell wall. A capsule is the dense, well-organized glycocalyx outside the wall. If the material is loose and slimy, the colonies look really wet, then it's called a slime layer. Slime layers don't stain very well, but capsules stain pretty well. So one thing about colonies that form capsules, oftentimes they look wet, glistening. So this is our mix A plate, and it had Klebsiella and staph on it. The colonies that are more wet looking, glistening, those are the Klebsiella. There's 2 Klebsiella plates up here at the front that were turned in this evening. They can be used for capsule staining. And then there's Klebsiella on the slants in the racks. I think you guys have any of these. Well, anyway, we might have to steal one to put over there. So there's Klebsiella on the benches for staining also. Anyway, they're the slimy looking guys. We also have our Azotobacter and Pseudomonas plates. Now you guys did not streak these plates, but you did put your soil in these tubes on the shaker. Remember that whole business? So those plates have been sitting in the box. They've been growing since last Wednesday. And what I'm going to do is put them over on the bench next to the sink over there. So you will recover your Azotobacter plates and your Pseudomonas plates. The Azotobacter will often produce really slimy bacterial colonies. It's kind of hard to see these. They're nearly transparent on this plate. Sometimes they're so slimy that the plates are incubating auger side up, and that slime will drip into the lid. So you may see really slimy samples. We're going to do a capsule stain that is a combination of Klebsiella and Azotobacter, and hopefully you'll get to see good capsules and hopefully slime layers too. Now, the capsule stain is a combination direct and indirect, kind of like the Dorner method endospore stain. The bacterial cells are going to be fixed, but it's not going to be a heat fix, it's going to be a chemical fix. So we're going to stick them onto glass with ethanol. We're going to stain them with crystal violet, and then the capsules will be visible around the cells as a clear layer. So it's going to look white. That means we have to stain the background in order to make the capsule visible. And again, you can stain the background either with Congo red or nigrosin. Oftentimes, people will show me their representation of a capsule stain, and what they've got is a bunch of purple bacteria on a white background. If the background is white, you cannot see capsules. Capsule is a white layer outside the cells. The only way you can see that is if the background is dark. So you have to make an indirect stain, a background stain. So here's the procedure for doing a capsule. You're going to make 2 small smears on one piece of glass. At one end, you're going to put bacteria from the Klebsiella slant and also from the Azotobacter plate. Try not to put too much bacteria on, and you're not supposed to mix these with water. You just put some bacteria on the slide. At the other end, you're going to put your morphological unknown. So a mixture of Klebsiella and something from the Azotobacter plate at one end. And I'll put this on the board. Morphological unknown is at the other end. Then you're going to put either nigrosin or Congo red into that smear. So there you see the 2 smears. The morphological unknown is a U shape. While that smear material is still wet, I'm putting nigrosin in. Again, if you're using nigrosin, the background should be fairly dark gray. If it's too light, it's really hard to see the cells or see the capsules against the background. You also don't want it solid and black, so don't use a drop of nigrosin, use your lube. Remember, sterilize your lube before you pick up the nigrosin, because that's an aqueous solution and we don't want cells growing inside. If this were a Congo red prep, the Congo red is red. So when you put it in the mix, it's not going to look black like that, it's going to look red. Anyway, then you let these dry thoroughly, and then you're going to fix, but you're not going to do a heat fix. So you're not going to pass these slides through the flame. Instead, you're going to fix with ethanol. If you are using nigrosin, fix with ethanol, so you have 95% ethanol in the green trays. If you choose to use Congo Red as your background material, when you do the alcohol fix, use acid alcohol. This is from our acid fast stain. So this has 5% hydrochloric acid in it. And the reason that I want you to use acid alcohol is because the acid is going to cause the Congo Red to turn blue, make it dark. You don't want it to be red, you want it to turn dark, which it will. When you put this alcohol on, you want to flood the smear because you have to leave it sit there for 2 minutes. So you want enough alcohol on there, whether it's acid alcohol or just ethanol. You don't want it to evaporate. So put a lot of it on there. It's going to stand up on top of the glass, look very wet. So there you see a flood of liquid sitting on the glass surface. That's supposed to sit there for at least 2 minutes, and then you dump it off. Don't rinse with water in between, so it stays on 2 minutes. You dump it off, and then put Crystal Violet on in place. Don't rinse in between because it will take the color off the slide. When you put the Crystal Violet on, you're supposed to let that stay on there for 2 minutes. This is probably potentially messy because it's hard to pick that slide up without getting Crystal Violet on your thumbs, your fingers. Anyway, so you let that stay on there, and then you're going to dump that off and rinse with tap water. When you rinse gently. So let the water coming out of these taps tends to come out fairly hard. So I recommend that you run the water across your fingertips and let that water then go on the slide. So it rinses it off, but you don't want to knock that background off. So this is what it looks like. Actually, I don't know if this was a Nigerson prep or if it was a Congo Red, because the Congo Red turns blue like that when you put acid alcohol on it. So you're supposed to look at these. You can kind of see what region of the slide you should try to observe. You want to find a region of the slide where the background is pretty uniform. On the one on the left, it says smooth, but what you're looking for is something that's fairly uniform, not broken up with big chunks and white areas. Again, you can't look at cells against a white background and see capsules. You have to have a dark background. You have to look around, focus with your 10x, look for an area that's nice and smooth, and then increase your magnification to 100x. What you see on the left is an illustration. Those are really big capsules, easy to see. What you see on the right is what you actually see. The capsules are tiny. Well, the bacteria are tiny in the first place. Can you see the capsule? White halo, white halo. Sometimes capsules, if you have cells that are in what we call a tetrad, where you have 4 cells in a group, sometimes the capsule will be on the outside of the tetrad. Consequently, if the cells break apart, you'll see a capsule that's only on one side. They look like cartoon eyeballs with this white area just off to one side. Anyway, so you're supposed to record, does your culture have capsules or does it not? Some of them don't. The other thing you're supposed to observe tonight is the flagellar stain. These are prepared slides, so they're in the slide boxes. We will not do flagellar stains. It takes too long and the reagents that are used are toxic, so we're not using them. In your slide box, you should have a slide that says bacterial combination or flagellar combination. It has one little smear that has Paratrichus flagella and one that has Amphitrichus flagella. Please remember, slides are expensive. Share them nicely. The Paratrichus flagellar preparation, you have to look at this with your oil immersion objective, otherwise you can't see them. So here you see a cell, the flagella. Notice how they're coming off in all directions. Paratrichus, paranemes around, the flagella are all around the surface. If they're Amphitrichus, and you can see the Amphitrichus flagella pretty well with a 45x objective. These cells are a lot bigger. Amphitrichus, the flagella are coming off the ends. Now, since we will not be doing flagellar stains with your morphological unknown, if you want to know, can your culture produce flagella, the best way to do so is to see if they can swim. So we're going to do a wet mount and see if your bacteria swim. If they swim, they have flagella. because bacteria don't make silk. If they swim, they're flagellated. If they don't swim, well, they probably aren't flagellated. On the other hand, they might just be lazy. We do have some pretty lazy bacteria. Anyway, so we're going to do this business. To make a wet mount, and you want to put a drop of deionized water on the slide. The deionized water is in this little plastic gizmo. So put a drop of that. Pick up then a visible amount of your bacteria and put that visible blob right in the middle of the water drop. Then you're going to take a cover slip, and instead of tipping the cover slip and try to push the bubbles out from under the slide, hold it parallel to the slide surface and drop it. So when it goes down, hopefully you will catch bubbles under the cover slip. You want to find a bubble because you want to focus on the bubble edge. So here you can see air bubbles under this cover slip. So they just look like this. There's three bubbles in there. You want to put that bubble in the center of your viewing field and begin focusing. So when you do a 10x observation, you can see the bubble edge as a really dark line. And then, of course, you have to figure out where are the bacteria relative to that bubble edge. This is inside the bubble. There are no bacteria in there. The bacteria are out here. You want to focus right on that bubble edge because oftentimes if you're going to see motility, it'll be right in here next to that bubble. This is showing 40x. The bacteria were motile, unfortunately, on a still image. That just comes out blurry. But if they're swimming, you'll see them going like the movie showed them in all different directions. If they're all going in the same direction, that's not motility. That's they're just going with the water flow. Bacteria don't swim in formation. If they're just shaking, that's Brownian motion. Bacteria can be hit by water molecules and they bounce, but they're not going anywhere. You see them swimming, that's motility. If they're flipping over and over, that's motility. This is called tumble. This is a run. Anyway, so the completion of your morphological unknown. You have to complete the morphological unknown report form, which is page 95-96. It has space for recording colony morphology. It has space for recording cell morphologies. And for each aspect, the stain that you were doing, you are supposed to record data, which is what you see, result, which is plus or minus, or acid-fast, non-acid fast, and conclusion. If it's gram stain, gram positive, thick peptidoglycan, no outer membrane. If it's gram negative, thin peptidoglycan, plus an outer membrane. If it's an acid-fast stain, and the acid-fast is red, your conclusion is these cells have high levels of mycolic acid in their walls. So don't repeat results in the conclusion section. You're also supposed to determine the identity of your culture by going to NCBI. This is going to require that you get a computer, and then on our web page, there's the currently up as of this afternoon, Morphological Unknown Identification Assistance page, and it has a grid. Each of you has a number that your morphological unknown is assigned, 1 through 30, and next to that there's a number. It's a letter number combination. It's called an accession number. You have to copy that accession number and go to NCBI, which is the National Center for Biotechnology Information. You look in the lab manual, it tells you what to do. And actually, if you look on the web page, it tells you what to do. Anyway, you record your accession number, you go to nucleotide under NCBI, and then put that number in their little search window and press return, and then it will open a page that tells you what your culture is. One thing to be aware of, some of the species names that are posted in here are not the same as the species names that you have. Use the ones from the web page. Please don't use the ones that are up here because these change sometimes from semester to semester. Also, don't assume that somebody else did a better job of recording morphology. Do it yourself because they might be wrong. So what do you have to turn in? You're going to turn in the page, the report form, front and back. You're going to turn in one full color illustration page that has all 5 stains. Gram stain, indirect stain, acid-fast stain, malachite green endoscore stain, and tonight's capsule stain. and it needs to be colored. Please use 8.5 by 11 white paper, no lines. If you guys don't have some, we have some. The department has some. I can go get it for you. It used to be over there on the counter. It's not there anymore.
I think it is. I think my backpack's just there.
Oh, it's in Anyway, you can look at the kinds of pictures that people have put up. Sometimes they make circles to put the images in. You don't have to. I've seen them not with circles. But you have to have all 5 images on one side, please. And you're going to turn in 1 properly streaked plate. So look at your morphological unknown plates. We've had them open a lot because you've been taking samples out to do things. So if they have been contaminated, restreak. And when you turn in that plate, it has to be a pure culture, it has to have well-isolated colonies, and it has to be properly labeled, which in this case is the correct technical name for your morphological unknown. You can't call it morphological unknown number 24. It has to have a name, which means you have to go to NCBI and find out what it is before you label that plate. Okay.
So what day are we turning this in on?
So I don't have a hard and fast due date for these. My recommendation would be not this week, next week probably, but I don't know what your circumstances are and how much time you guys have and how many other classes you're trying to take and how much you work, etc. So I don't put hard and fast due dates. I encourage you to try to get this done as quickly as possible. Get it out of your hair because we will start the next unknown before you know it. Okay?
Thank you.
Yep. You're going to see students from the other classes in here in an absolute panic attack because they have a deadline. They have to have them done. I just say, well, you know, get them done. Try to get them done before the semester ends, okay? I've actually had these turned in like the week before the semester ended. But it gets tricky because your cultures die and then you have to go back into the tube and then they get contaminated. It's like, just get it done. Okay, so I'm going to put the information on the board, how to do a capsule stain.
Press that button and then all.
I'm just more concerned with gotta make sure I get my pictures.
Do you ever care? Would you take the red pill or the blue pill?
I don't think I would.
So you want to take that blue pill? This is all very important.
Oh, tap the screen like on the green because it'll be.
Okay.
Oh, yeah, they are. Are they off the webpage?
Oh no, I don't know where that's from, but it's the, I don't know.
Oh, maybe we just do that and she puts it up.
I'm gonna bring it like as much as I can because I'm trying to the first minute we're talking.
Oh, I had to for two.
I put lots of lotion on my skin today, and you can see it all over the bench. And it goes.
How far did you get on this? Just this stuff?
Yeah.
Like
I haven't filled any of my skin on this paper throughout all the labs. I just need to transfer all my
I didn't draw any of my shoulder.
I just took pictures.
I did too, but now I have to figure out which ones
I've
are
been
so
drawing it, but
Look at you. Look at you. Wait, but that's if we know what it is.
Two weapons and two
yeah, so I
I hope they remember that whole thing.
Yeah.
What?
I don't know, they said it.
She's not, she's.
They go with one
But it's just directions.
I know.
You don't need to record anything.
It's a 26-minute fucking
Yeah, that's my second one.
I don't care, I'm just gonna.
No, we don't need that much stuff.
What they want.
You're gonna waste my time.
If they want it,
Oh, so we got to the chance. Yeah. Well, we're gonna agree.
Okay, can I have your attention briefly? The plates you need for your Zodavector and Pseudomonas are back here on the side bench. Please pick these up. This is part of the prokaryotic cell introduction we're doing tonight, but you need the Zodavector plates, which are the white ones, for your mix for the capsule stage. So please pick those up. The Klebsiella is in slants. They're on the table, so they're in the green tray.
So we don't mix that with water?
Yeah, we don't.
You just got air conditioners.
Where do we get that Azoto back?
Is it there, or is it in our, is it the stuff we're picking up here?
They can't mind their own business.
You guys don't have anything else.
Some stuff, but we got the house together, you don't have supplies, yeah, we're moving a lot of stuff.
You don't have to, because you're gonna put something on here. Either nitrogen or carbon. You just gotta part it around there and it's wrong.
pictures have anything good in it?
On this one,
And I've actually
Ooh,
seen
is it
pretty
slimy?
good capsule
Really
stains,
slimy.
even though you put water in. If you put them in water, just don't stir them around a lot. You can put just water to get them off the loose. And then once you
we
put
dumped
it in,
it in.
then kind of stir it a little bit. You're just not supposed to stir them because the capsules can contain.
Klebsiella
Capsules
and
usually
Azoto.
don't that much, slime like.
So that's
Oh,
right
and
here.
then some bar.
did your other one do anything cool?
No, they both did really do. This one just kind of looks wet. This one just looks like that. Nothing hardly.
Barely did anything.
Yeah, it's
My
weird.
gas is still working. Makes me puke when I smell it.
God, that drawer stinks.
Oh, yeah. Oh. That smell is just too much for me. Like, it's one of the first things I've smelled in like a week.
Oh God. Yeah, we're looking for that.
So we're mixing the Klebsiella. You're welcome to
Mine's
the
doing that
fire.
too.
Do you know where they put Azoto back from?
Is it the enrichment we
She
just
said
picked
there
up?
was the really slimy.
One we just picked up.
This
Or
one, right?
soil sample.
Yeah. Right? The soil sample was the
I believe so.
Azoto.
So I think that's the
She's-
ADM, right?
Okay.
She did the streaking
I'm
for
just
us.
thinking.
Are they in the back?
Sure. I'm gonna double check.
Yes, there are over on that thing.
Really good idea. Oh, we're starting? We started walking each other. We go into that.
I know. Oh, we're starting.
Well, you want it out. If you want. Yeah. It might not even say, but I know.
I'm just gonna throw the whole shit out.
AEM, right?
Okay, he's gonna double it.
Azotobacter enrichment medium, Pseudomonas enrichment medium. Yeah.
There's really slimy. Be careful, there's like
We're all crossing.
Dude, it's so
Oh,
His is like
is yours real juicy?
She said use this.
All of us
Be
use
really
that?
careful with these arrangement. You're doing an acid fast. The malachite green N-spore stain, the instructions are on page 78, not page 80. It is the malachite green stain because the Dorner method was not your unknown. And make sure that you're looking at your unknown as opposed to the bacillus, which is at the other end of the slide. And then the last stain is the capsule stain that we're doing tonight. That is on page 80. Again, what I want you to do is finish the stain for your capsule stain and then stop. Don't get the microscopes out. I'm going to introduce prokaryotic cells and then you will do another stain, which is a gram stain, and then you can look at capsule stain, gram stain, flagella, etc.
She wanna
Do your wet
one.
mount. You don't want to do wet mounts until you have your microscopes out.
I thought that looked good. Damn, this shit's hard.
Lots better. Oh, yeah, and it dried a lot faster, didn't it? That's why you don't use dry. It's funny how that
Do I pause it?
It's possible.
It's not.
It's been recording this whole time.
No, I just turned it on a second ago.
This is particularly important when you do the capsule stain. Remember, capsules are outside the cell. So if your cell is a certain size in the indirect, the cell should still be the same size in the capsule. The capsule is outside, if they have one, and they may not.
I have. I took a picture of some.
I know, but I didn't label
I did. Somewhat.
So I know the lab manual suggests that you
I know one
should
of them
keep
I can't see
measuring
'cause
these
it's
with
blue.
every stain. I recommend that you not. Measure them once with the indirect. It's right. The rest of them, the cells are heat fixed and they shrink.
Oh, thank you.
Yeah, yeah.
Are we keeping these? The soil plates?
Yes. No, here.
Sitting in the middle of the back.
Oh.
That one. Never get near.
Yeah, if anything, Nathan was like the best one.
I know, I sat there.
Yeah, and they both like that part of
Okay, if you could kindly settle and stop talking so that we can get through the introduction to prokaryotes. I realize that you've been introduced to prokaryotes a little bit because we've been doing all these stain techniques, and they all involve bacteria. So you did your gram stain, you saw bacteria on your cheek cells with the direct stain, you did an indirect of your morphological unknown, acid fast, endospore stain, etc. Tonight we're going to be looking at the prokaryotes just a little bit more. Again, they are the bacteria and the archaea. Bad news, good news, depending on how you're looking at it, we don't have any archaea in the lab. We don't actually get to work with archaea, we're only working with bacteria. As a group, the prokaryotic organisms are the most ecologically and metabolically diverse life forms on the planet, and we know really relatively little about them. Please stop talking. I'm trying to present information. So the term prokaryote, the word means before a nucleus, and this term was actually introduced in 1937, but it was Roger Stanier and his associates looking at prokaryotic cells with electron microscopes that actually defined what was going on. Those cells do not have a nucleus. They don't have material surrounded by a nuclear membrane. This wasn't determined until 1961. A real definition of what do we mean by prokaryotic cells. So the Burgess Manual of Determinative Bacteriology, as I said, this is our reference for bacteria. It was first published in 1923. It provided identification schemes, ways to identify bacteria, and became a very important reference. It's been redone numerous times. In this laboratory, the oldest Burgess Manual that we have is the 8th edition. It looks like that. It's a very ragged book. It was published in 1978. And it divided the prokaryotes into bacteria and cyanobacteria, as though those were the two really different groups. Turns out that's not the case. In 1989, this set of Burgess manuals was finally published. It was completed. This is the 4-volume set that has volume 1 is ordinary gram-negative, volume 2 is ordinary gram-positive, volume 3 was bacteria with unusual properties, and volume 4 was filamentous bacteria. It was like, okay, now we've got the bacteria sorted out. Turned out that thing was completely inaccurate, so it had to be redone. In this laboratory, these two volumes of that set, volume 1 and 2, because they were the ordinary gram-negatives and ordinary gram-positives, these were used extensively by students, so you'll notice that the back of the book is held together by duct tape. And they're very useful, but unfortunately not terribly accurate anymore, simply because we've learned so much more about bacteria and there's many more groups. So technological advances, primarily in gene amplification and nucleotide sequencing, and now what we call metagenomics, which is sequencing from the environment, looking at whole communities. We know that there's a lot more to the prokaryotes than we see in those volumes. People are still working on trying to unravel the phylogeny, the evolutionary history of prokaryotic cells. So we had another set, the Burgess Manual, the 2nd edition of the Systematic Bacteriology. This one was supposed to be 7 volumes. I think there are actually 7 books. If I brought these out here, so 1 volume, 1 book, and then the next one 4 books, and then the next one 7 books, you kind of get the idea. the volumes of information are growing significantly. Anyway, then once it was determined that the archaea were significant, then those guys had to have their own representation. So they're in one of the books in this new set. The proteobacteria are represented by 3 books. The Firmicutes are 1 book, and then you have 1 book that has spirochetes and bacteroidetes and chlamydia and some other things. And then there's supposed to be a 2-part book for the actinobacteria. Well, we have part A, and as far as I know, part B has never been published. So what happened with the Burgess manual, there has been a movement to put this thing online. And people are saying you can't keep up. Every time you publish this manual, it's out of date. And it's so slow to get it published. So finally now, I think just last year, the Burgess manual is now online. And it's called the Burgess Manual of Systematics of Archaea and Bacteria, or the BMSAB. We do have access to it. It's going to be updated, I don't think quarterly, I think once a year they get an update. So they're struggling to keep up with the information because new information is coming so fast. Well, with that much information about prokaryotes, there's no way that you get to be introduced to everything. So you just get a smattering in this lab. You have the morphological unknowns. As I said, there's 30 of you in here, 30 different morphological unknowns. You can see images. Well, there aren't 30 up here, I don't think. So some of them are missing. Actually, I don't know how many are up there. So that's one part of the introduction. Each one of you has a different type. Your goal, as I said, is to learn about stain techniques, learn about aseptic technique, learn about colonies, etc. The other thing that we did, which is part of tonight's exercise, was the enrichment procedure. So we put dirt in tubes. The tubes are still sitting over there on the shaker. And then 1 tube was put into a boiling water bath. So we want to talk about that for a bit. This is another assignment. So you've got the morphological unknown assignment, which is 30 points. You also have an enrichment assignment, which is 20 points. So enrichment, this involves using media or special techniques. What you're trying to do is modify circumstances or modify the environment so that you promote the growth of certain organisms and inhibit the growth of other organisms. Enrichments are not pure cultures. So this question, have we specifically designed media and one unique enrichment technique? And the answer is yes. So the enrichment media are on the shaker. Red caps, blue caps. That's your Pseudomonas enrichment medium. And then we had a boiling beaker up here that we pasteurized a soil sample in. So that was our technique. Defined media. Remember, they have all the nutrients in specified quantities and you know exactly what's in there. These are our 2 enrichment media. The plates that you picked up tonight are labeled PEM or AEM. PEM is Pseudomonas enrichment medium. Pseudomonas enrichment is on the left. AEM is Azotobacter enrichment. That's what was in the broth. The broth had no agar, so it's just liquid. It's the same material that's in the plates. The only difference between the plates and the broth is that the plates have agar in them. It's a solidifying agent. So our Pseudomonas enrichment medium, sodium benzoate, is our only carbon source, and it's toxic. I put this on the board. Why is it an enrichment? Because sodium benzoate is going to inhibit the growth of most bacteria. It's used as a preservative. It's toxic. Most bacteria can't use it as a food source, but some pseudomonads can. So if they can use sodium benzoate as their carbon source, they've got their nitrogen as an inorganic form. It's got ammonia in here. It's got a bunch of other minerals. It's got a buffer. It's got water. They're all set. The Azotobacter medium has mannitol as a carbon source. It's got some other minerals in here. It's got buffers. There is no nitrogen in that medium. So the AEM, the Azotobacter enrichment medium, also says on the plate nitrogen free. That's the white one. So there is no nitrogen in there. We're selecting for organisms that can fix nitrogen. They have to be able to pull it from the air to grow. Okay, so yes, we did use those special media. They were on the shaker for about a week. Then they were taken off and put onto the plates. Again, the only difference between the broth media and the plate media is agar. I have students write their enrichment description and they tell me that the organisms were put on nutrient agar. No, they weren't. They were put on Pseudomonas enrichment medium or Azotobacter enrichment medium. Broth first and then plates. So they look like this. The Azotobacter one's white, the Pseudomonas one is tan. They've been growing since last Wednesday. Again, Pseudomonas uses sodium benzoate, it's the only carbon source in there. There are quite a number of Pseudomonads that can use that. The reason we put them on the shaker initially is because Pseudomonas are respiratory, they want a good supply of oxygen, so they were mixing in there. They can use sodium benzoate, and organisms that can't are not going to grow. But there's a question asked there, do you think the colonies here are made by Pseudomonas? Well, probably, but is it guaranteed? Do you think Pseudomonas is the only type of bacteria that can use sodium benzoate? Probably not. So that culture that's growing on that Pseudomonas plate is definitely not a pure culture. So can we be certain they're Pseudomonas? No. Consequently, this is a potential start point for a semester project. A lot of you are looking at air plates, but this is another possible start point. There's something on that plate. It's probably Pseudomonas. Pseudomonas is a very big genus, many different species. So again, potential start point. The Azotobacter medium, again, lacks nitrogen, so only nitrogen fixers can grow on that medium. Azotobacter can, but do you think Azotobacter is the only organism type that can fix nitrogen? No. In lecture tonight I said Rhizobium can, Agrobacterium can. There's a bunch of bacteria that can fix nitrogen. So again, is that, it's not a pure culture, and do we think those are Azotobacter? Maybe, but we cannot be certain. So again, potential start point for semester project. You want to find out who was in that soil sample that you brought in. It might be Azotobacter and it might not. The other thing we did was an enrichment for bacillus. The bacillus organisms are endospore formers, so we put soil into some water and then we put it in a boiling water bath for 1 minute. This was pasteurization. The pasteurization process will kill vegetative cells, but it will not kill spores, endospores or exospores. So then what we did is took a sample of that liquid and put it on an agar plate. I don't know where those plates are, because you guys were supposed to put them in your lab drawers. Hopefully they're in there somewhere. So we're pretty sure that what's growing on there is something that makes spores. But now, since bacillus has been divided into all these categories, we put them on that plate. Do you think it's only one species of bacillus? Well, look at the variation in morphology. You could say, no, there's definitely more than one thing on there, and they might not even be bacillus. They could be painibacillus, lysine-bacillus, brevibacillus. So again, potential start point for a semester project. We don't really know what's on there. So, the other thing people have been showing me tonight is their air plates. Air plates are a real good sample. Well, air's a nice habitat. When you're thinking about your hypothesis, where did organisms come from if they're in the air? Probably off of human skin, off of clothing, off of pets. If you have, if you took your sample in a house, you had carpeting and pets come in, they walk across the carpet and the carpet pulls things off their feet and flings it into the air. So you want to address your hypothesis to what kinds of things do you think might have gotten onto that air plate. They can be coming off of skin surfaces, they can be coming off of soil, they can be coming out of water. Anyway, a lot of possibilities on there. So that's another, just try to stay away from the fungi. Don't grow fungi. So you have lots of different types of bacteria. Good start point for semester project. So a lot of people are thinking about starting their semester projects tonight. That's good. Now, the exercise for the enrichment. This is a 25-point assignment. I think I said 20 a minute ago, but it's 25. What I want you to do is prepare gram stains from the enrichment cultures that we did. Well, there's 3 plates now. Pseudomonas, Azotobacter, and Bacillus. On that slide, you do a gram stain, you've got 3 smears, Pseudomonas, Azotobacter, Bacillus. You can do 1 more smear, take it off of your air plate, especially if you want to start your semester project from your air plate. Put 1 more smear up there and get a chance to look at the culture. So you're going to do a gram stain. you're gonna write a one-page report where you name the organisms we were enriching for. The organisms are Pseudomonas, Azotobacter, and Bacillus. You explain what did we do for the enrichment. I just went through this and I had written it on the board earlier. And of course it's in the lab syllabus. Pseudomonas, we use sodium benzoate. Why? Because it's toxic. It kills most bacteria. They can't use it as a food source, but Pseudomonas can because they can use a wide range of unusual organic compounds. The Azotobacter medium has no nitrogen in it. Why? Because Azotobacter can fix nitrogen. They can take molecular nitrogen from the air, use it to make nitrate, nitrite, and use that to make organic compounds. The Bacillus, we pasteurize them. Why? Because they make endospores. Endospores are thermoduric. They're resistant to heat. So they survive. Pseudomonas that was in there, Azotobacter that was in there, whatever's in there dies. The Bacillus survive. We put them on nutrient armor. Okay? So you're going to turn in a written information. You're supposed to do a full color illustration. Well, you can just put that on the same page. Just draw Pseudomonas, Azotobacter, Bacillus. Draw them big enough you can see them. And 3 auger plates. The easy part of this is you didn't even have to streak 2 of them. But if you picked them up, and everybody had one over there, I know because I streaked 30 plates of each, Pseudomonas and Azotobacter. And then you just have to put your Bacillus plate with that. So you're turning in written information, illustration, plates. Now, the last thing we're looking at tonight is Introduction to Cyanobacteria. This is a phylum. They're in the lab manual as a separate group because people used to think they were a big deal, cyanobacteria. Now we know they're just bacteria. They are bigger than some others. And here you can see them being sold in health food stores. Spirulina is the most commonly used as a food supplement or dietary supplement. They're oxygenic phototrophs. They're gram-negative, but some of them form sheaths, some of them form capsules. Some form heterocysts, some form akinets. This is the first set of organisms where you are going to be expected to identify specific organisms on site and be able to name them. And also give me the taxonomy. Well, the taxonomy is, they're in the domain bacteria, phylum Cyanobacteria. There is no kingdom. And there are 5 genera. So you're just going to be looking at the prepared slides. They're in the boxes. They're also on the website. I don't know if we have any material to do wet mounts from. I'm not going to bring the wet mounts in tonight anyway. We'll bring them in on Thursday. So I just want to go through the slides. So this becomes very repetitive. Domain bacteria, phylum Cyanobacteria. There is no kingdom. The first example is Anabaena flossaqui. Anabaena is a very common genus. Flossaqui is a particular species. This is a helical mutant, and the cells are artificially colored. What you see is chains of cells. A chain is a trichome. Heterocysts show as enlarged cells. This particular organism type is studied rather extensively as a potential food source. Anabaena. The next one is still domain bacteria, phylum Cyanobacteria. This is nostoc. Nostoc can live in colonies that can be this big around. The cells are microscopic, but they form a glycocalyx around them, and the cells live inside. So what we're looking at is the edge of a glycocalyx, and inside are these chains of cells. They look somewhat like Anabaena, except they're in this glycocalyx, which Anabaena is not. So that's nostoc. The next one, domain bacteria, phylum Cyanobacteria. This is Oscillatoria. Oscillatoria are like Neckos or Smarties. The ASM group used to give away Smarties, and we called them Oscillatoria. So the cellophane wrapping on Smarties is the sheath that these guys live in. The disc-shaped Smarties. Y'all know what Smarties are, yeah? Yes. Okay, so the disc-shaped candies are the individual cells of the cyanobacteria. you can see the cell margins as dark lines here. So this is a stack of cells inside of a sheet, like Smarties in a pack, except the pack is way long. And they get their name, oscillatoria, because they move and twist and slide around. They do not have cilia, they do not have flagella. Apparently cytoplasmic streaming somehow allows them to do this movement. So that's oscillatoria. The next example is Gleocapsa, so domain bacteria, phylum Cyanobacteria. Gleocapsa, these cells live in a capsule. Sometimes there's one cell per capsule, as you see here. Sometimes two, sometimes three. Again, this is artificial color. So if you see them live, they're green. The other thing about, remember I said glycocalyx is sticky, so it serves as an attachment mechanism. The slide that has Gleocapsa on it has a bunch of other stuff stuck to it. So there's some weird diatoms in there that look like shell macaroni. There's a bunch of weird stuff, so you have to look around and find the Gleocapsa. And then our last example, domain bacteria, phylum Cyanobacteria, spirulina. These are the spiral bacteria that are usually sold in health food stores. And I'm not sure what the status of our slides are. We had these slides that were purchased and they had beautiful spirulina on them, and then the color all faded away and you couldn't see anything. And I believe some new ones were purchased, but I have one like that for lab test purposes. Anyway, so what I want you to do before you start looking at prepared slides is do a gram stain. A gram stain should take you what? 10 minutes max? Dream on. So do a gram stain, Azotobacter, Pseudomonas, Bacillus, and something from your airplane. Can I erase this one?
Let me take a.
And then, as I said, if you don't have time tonight to look at everything, Thursday, that's all we're doing is looking.
Perfect.
We're not doing any more stains, we're just looking.
Thursday's like kind of like an open lab.
Yeah,
deal.
Thursday's gonna be very much like an open lab time.
Love that.
Yeah. I mean, we have to go through the presentation on fungi, but then I'll do it again.
Okay.
This one Bacillus.
You're gonna give us a break?
Yeah,
You're so nice.
yeah, for several labs now, we get a break.
Look at the Bacillus, bro.
Move my bacillus, bro.
Decolorize. Watch the color move, and when it stops moving, you rinse. Saffron, 60 seconds. Rinse, air dry. You're done. Yeah, 60 seconds, 60 seconds.